DNA is something all of sci-fi love to talk about beyond the AI takeover. Actually, I wouldn't know. I don't really watch sci-fi. But how well do you understand DNA? No, really. What do you know about DNA? The majority of people don't even know that what it stands for. Maybe someone who took regular honors bio will say something like, it stores your genetic information. Well done. Okay. DNA is so much more significant than one can ever imagine. Buckle up because we're going to talk about the one thing I loved in AP Bio. Alright. The central dogma of molecular biology is the process of DNA creating protein. It goes as follows: DNA, transcription, RNA, translation, and protein. Let's start from the basics. What's the difference in DNA and RNA? DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. The difference in name is due to the sugar in its nucleotide, the smallest form for these nucleic acids. DNA is double-stranded. That's why DNA looks the way it does. RNA is single-stranded, so keep that in mind when you're watching those movies when they randomly bring up pictures and claim that all of it is DNA, when clearly it's not. The most important difference for our discussion has to be the nitrogenous bases. Those are the colored parts in the middle of DNA. Both RNA and DNA have adenine, guanine, and cytosine. DNA has thymine, and RNA has uracil. Thymine and uracil both bond to adenine, while guanine and cytosine slowly bond to each other. Based on the model, we go from DNA to RNA. This is done by transcription. I'm not going to go into specifics, join AP Bio for that, but I will explain the 3 steps of this process. First, step 1 is initiation. Enzymes uncoil DNA, because DNA is coiled around a protein known as a histone, and unzip, aka break the hydrogen bonds between nitrogenous bases. From there, RNA polymerase connects to the template DNA strand because of something known as the promoted region. Remember this for later. And RNA nucleotides begin to connect to DNA. Next is elongation. As the name suggests, we are elongating the RNA strand by adding more RNA to it. We end with termination. Very self-explanatory. Now we got our mRNA, the RNA produced by transcription used to send the protein code to the ribosome. But we are not done yet. If I were a prokaryote, it wouldn't matter. But the last time I checked, I'm not a simple microscopic organism. This process is very important in order to produce the right protein. Splicing. Splicing occurs in order to remove the unnecessary sections of mRNA, called introns, from the necessary sections, called exons. Why this is so important is because you can use the same mRNA to produce different types of protein. In order for the mRNA to actually survive long enough to go to the ribosome, we have to add a GTP cap and a poly A tail. Now we have a mature mRNA. Hip hip hooray! Oh my gosh. Now our big boy mRNA needs to actually do its job and give us protein. So it travels out of the nucleus and into the ribosome. From here, we begin translation. Translation simply is converting the mRNA's code to protein chain. Like transcription, translation also has 3 steps. Step 1 is initiation. The mRNA binds to the ribosome using the start codon. 3 nitrogenous bases equal 1 codon, and a codon equals to an amino acid, the smallest form of a protein. The start codon brings the start amino acid, the thymine. Step 2, elongation. tRNA, which are RNA molecules that carry amino acids, bind to mRNA in the ribosome, and the ribosome binds to the amino acid. tRNA carries to methionine and begins the protein chain. This keeps going until we reach the final step, termination. The ribosome reads the stop codon, which does not have an amino acid, and releases the protein chain, plus disattaches mRNA from the ribosome. Now we have reached the end of the process and we have made our protein. That was a lot of yapping for a whole lot of nothing, but I assure you that everything I am about to say moving forward is connected to this process. Everything. Y'all ever heard about adaptation? I bet y'all haven't. It's not like every science class for every year you are in school has at one point talked about adaptations. That doesn't happen. Okay, enough of that. Adaptations come from mutations. Not all mutations, however, are adaptation. A mutation is a change in the DNA sequence of an organism. Thank Daniel A. Gilchrist for that definition. Now, most of the time, our mutations are dealt with naturally in our body, but that doesn't mean we can't get them. Now, notice in our definition, we mentioned DNA sequence. The first step in the central dogma is DNA. Therefore, if a mutation makes it through the entire process, it becomes part of our genetic system. For this, I'll be using the hypothetical example of the J. Jimmy John species in order for this to make sense. J. Jimmy Johns live in the wonderful region of Siberia. They are furry in nature, small in physique, and have large stompers in order to mark their territory. One day, two Jimmy Johns give birth to a defective Jimmy John. Instead of large feet Jimmy Johns are known for, this Jimmy John has small, tiny feet. It was due to a mutation in the Hox genes that changed the Jimmy John's body to have small feet. Going out of the example, mutations are random, as seen here. They can occur at any time. Hox genes specifically code proteins that are related to the body structure. For this mutant Jimmy John, a particular Hox gene had a mutation and went through the entire process of protein synthesis to create the small feet it has now. Just one mutation caused a massive change in physique. Now let's see what happens next. This Jimmy John grew up healthy and, to most of the surprise of the Jimmy John species, was a nimble freak. This Jimmy John moved much faster than the average J. Jimmy John, but due to its small feet, it could hardly attract a mate. It got so bad that it left the pack and moved up north where the forest is thicker and trees. However, this Jimmy John thrived. The small stature and faster speed made it possible for this Jimmy John to adapt quick, and eventually found a Jimmy John pack where it's found its mate. The Jimmy John was far better suited for the environment over the average Jimmy John, and often was the highest scavenger of the pack. Over time, this Jimmy John reproduced, and his offspring reproduced. More and more of this Jimmy John pack had smaller feet until it was a different species altogether. We now recognize the species as J. Little John. Adaptations are not random. Like I said before, all adaptations are mutations, but not all mutations are adaptations. The small feet adaptation that Little John had received was a result of a mutation, and that mutation created an entire species. That is the power of DNA and the central dogma of molecular biology. Other examples that we are more familiar with of mutations that result in adaptations are mimicry and camouflage. They mostly occur due to mutations that are coded for fur or skin to change in color and resulted beneficial to them. There is another way to affect protein synthesis: gene expression and regulation. Gene expression is pretty self-explanatory. Genes are either active or not. Regulation is controlling whether a gene is on or off. Now we know the dogma by heart, I hope. DNA, transcription, RNA, translation, protein. Using regulation, the gene either allows RNA polymerase to transcribe the DNA or it is permitted. Let's see this in action with the J. Jimmy Johns. This particular pack of J. Jimmy Johns live in the Gobi Desert. During the colder parts of winter, these Jimmy Johns maintain their furry coat, of course, to stay warm. However, during summer, where the desert becomes too warm for fur, they become bald. This species of J. Jimmy John is called the J. hairless John for its ability to regulate the growth of its hair. Now, it doesn't really go bald, it just shortens greatly the amount of hair it grows. Let's take a closer look. Right here, we see some DNA. There is a small part of the DNA that specifically codes for hair growth. Another part of the same DNA, we see an enhancer. This enhancer, when combined with transcription factors and promoter, folds to allow the hair growth gene to activate. This is because DNA is long and coiled, so each part is on a different section of the DNA. Now, this part we are familiar with. Replication bubble opens, RNA polymerase begins to transcribe, and we know the rest. When the weather starts getting warmer, another gene for shedding gets activated, while the hair growth gene is off. Thus, a J. hairless John species is able to survive in the Gobi Desert. Not all is positive though. Certain disorders are caused because of protein synthesis malfunctioning. For example, type 1 diabetes, and honestly most autoimmune disease. But there isn't a lot known about autoimmune diseases. We can't make inferences based on what we do know. We can infer that due to a mutation in DNA that codes for the immune system, like antibodies, was transcribed and released, the error caused it to, for type 1 diabetes, to mistake insulin-producing cells as a danger to the body and destroy them. Another example is sickle cell anemia. What's that? This actually was an adaptation against malaria. Basically, DNA codes for blood cells to have a crescent shape instead of the circular shape it's known for. Then it gets released to the bloodstream. But the most important impact the central dogma of molecular biology makes is on biotechnology. In fact, the dogma is fundamental for biotechnology. Without understanding the process, we could have never done anything related to genes. We wouldn't have forensics to discover murder without gel electrophoresis. It's actually a lot more helpful, as it doesn't relate to DNA but also RNA and protein. It just depends on what you are looking for. We wouldn't be able to create particular medicine without, like insulin, without recombinant DNA technology. To do this, you insert DNA that codes for insulin into bacteria, and bacteria goes through protein synthesis and starts producing insulin to be used. Creating the amino acid chart was done by Marshall Nierenberg and Heinrich Matthei by taking RNA and producing amino acids. From there, they will change the codon to see what will be produced. Afterwards, they will make a chart with other scientists in order to show all combinations that produce the 20 amino acids. Even one of the largest modern science projects, the Human Genome Project, used the Dogma in order to discover all the genes to advance in molecular science. In conclusion, I really like the central dogma of molecular biology. It is super awesome and cool.